Hello, my name is Nuria Contreras and I am a translational researcher at the Allergy group from San Pablo-CEU University in Madrid, Spain. On behalf of all my co-authors, I will be presenting our last publication in Allergy entitled as Multiomic Integration Analysis for Monitoring Severe Asthma Treated with mepolizumab or omalizumab. Asthma prevalence and severity have progressively increased in the last decades, leading some patients to fail at achieving disease control with standard controller therapies. In such cases, biological treatments have become increasingly important for managing the disease. However, despite their proven efficacy, their broader molecular impact and time-dependent systemic effects remain poorly understood. Additionally, the lack of reliable biomarkers further limits their proper monitoring, hampering progress toward personalized medicine. Here, we aimed to investigate the immunometabolic effects exerted by mepolizumab and omalizumab to identify potential biomarkers for monitoring severe asthmatic patients. For this purpose, a total of 56 severe asthmatic patients were recruited from a Spanish hospital and treated with mepolizumab or omalizumab. Serum samples and different clinical parameters were collected before treatment initiation and then after 6 and 18 months of having started the treatment. Our findings show that both mepolizumab and omalizumab induced clinical improvement in these severe asthmatic patients. Moreover, after performing a targeted metabolomic and proteomic analysis, we observed that both treatments induce specific changes to both profiles that may be related to the specific point of blockade exerted by each treatment in the Th2 immune response. However, some metabolites and proteins, as happens with sphingosine-1-phosphate and also OLR1, were commonly changed by both treatments. Then we performed multiomic integration analysis of our results using Diablo and in parallel a ROC multivariate analysis to jointly obtain the best set of biomolecules that could act as potential biomarkers for monitoring each of these treatments. After both analyses, omalizumab did not show a clear clustering after performing multiomic integration and neither discriminant multivariate ROC curves. On the other hand, in the integration model for mepolizumab, we observed that baseline in orange is clearly differentiated from the other two time points, and that this separation is determined by a set of 8 biomolecules. In addition, the multivariate ROC analysis revealed a set of biomolecules with a selection frequency over the 80%. Finally, from the common set of biomolecules obtained from both analyses in the mepolizumab group, we also observed that they significantly correlated with improvements observed in the previously analyzed clinical parameters, highlighting their potential as biomarkers for monitoring severe asthma patients undergoing treatment with mepolizumab. Therefore, our results suggest that mepolizumab and omalizumab treatments induce different metabolic and protein profiles, as well as a clinical improvement in severe asthmatic patients. Also, that a set of biomolecules explains the differences exerted by mepolizumab treatment along time. And finally, that this set of biomolecules significantly correlates with the clinical variables suggesting its potential as biomarkers for mepolizumab monitoring. Thank you all for listening, and I encourage you to read our paper for more detailed information. I would also like to extend my gratitude to all the co-authors who contributed to this study. We will be happy to answer any questions you may have via email.